So, hello to everybody. Thanks for coming, for showing up. Today, we are very lucky to have our guest from Malta, Rosienne Farrugia, that will give us a seminar or webinar about new tools that we can use to explore genomic landscape. Rosienne is a senior lecturer in genetics, genomics, research methodology, and hematology within the Department of Applied Biomedical Science at the Center of Molecular Medicine and Biobanking. And her ongoing research interests focus on genomic of diseases, including variety of diseases, tetrahydrobiopterin deficiency, idiopathic hypogonadotropic hypogonadism, polycystic kidney disease, Parkinson's disease, and myocardial infarction. She's also part of AtheroNET. Previously, she was part of CardioRNA cost action. And she will enlighten us with new things and basic and advanced things about high throughput sequencing. So, Rosienne, without further ado, please give us as much as you can with the information on this topic.
So thank you for that introduction and good evening, everybody. Since I've been told that it's a mixed group of people, I started this at the very basic and kept it relatively simple. And because I'm a biologist, I will only stick to the technology and not the data handling. I'm sure that there will be other seminars in the future on data handling. So for the next hour or so, I will just take you through the journey of the development of high throughput sequencing, which nowadays is no longer such a very new thing, or at least the second generation sequencing, which most of us use on a regular basis, is now becoming something a little bit of old school stuff. But there are the up and coming third generation sequencing technologies, which open up further possibilities of research, especially when one is looking for structural variants. So with high throughput sequencing, what we do is we can get the entire genome. So here we go. Each cell has DNA, and we can get the entire DNA molecule. For humans, that's 3 billion nucleotides, and we can actually sequence that entire DNA molecule in a single experiment. This is something which is like relatively recent in the sense it's something of the last 15, 20 years. Before that, sequencing, what now we call first generation sequencing, was limited to dideoxy chain termination Sanger sequencing, which meant laboriously sequencing very short fragments of DNA per experiment. So basically, in Sanger sequencing, what we used to do was get our DNA, amplify parts of that DNA, and then sequence parts of that DNA, tiny small fragments of that DNA. The initial incarnation of Sanger sequencing, if you want to call it that, was extremely laborious because we had to prepare 4 different reactions for every fragment we wanted to sequence. Because Sanger sequencing and all types of sequencing really depend on incorporation of a single nucleotide and the ability to check what nucleotide has been incorporated in that sequencing. So at the very start, when Sanger sequencing was first put together in 1977 by Fred Sanger, what used to happen was that for every fragment, there would be 4 reactions run in parallel. Each reaction would have all the things necessary for PCR with the addition of these special nucleotides, which are called dideoxynucleotides. These are blocking nucleotides because they miss a 3' OH. Because there is no 3' OH, a single nucleotide is added and the reaction stops there. There is no way that the chain can continue elongating. And those nucleotides were labeled with radioactivity. And radioactivity looks all the same, whatever it is bound to. So you would need 4 separate reactions, one in which there is radioactive C, one in which there is radioactive G, one with radioactive A, and one with radioactive T. And those 4 reactions would run in parallel, and then the 4 reactions would be loaded onto 4 lanes of a sequencing gel, and electrophoresis would be carried out so that the fragments would separate. And the smaller fragments always move faster than the larger fragments. So such a gel would be read from the very bottom, and you can actually read the sequence of the fragments based on the radioactive signal developed onto an appropriate film. So for this particular fragment, if we start reading from the very bottom, we've got 4 A's and then a C, an A, a C, 2 A's, and then a T, and so on and so forth. And you can read the sequence. Now, the system, as you can imagine, has a number of limitations. It has the advantage that we can actually sequence parts of the DNA. But there are a number of limitations. Amongst them, that you need to know the sequence, because your standard mix for PCR requires the use of primers, which are specific to your sequence, and you can only design those if you know the sequence. Sequencing was limited to very short fragments. You can sequence 300 base pairs in one run if you have a very good gel. It was laborious because you needed to run 4 parallel reactions per 300 base pair fragment that you're sequencing. And we were using radioactively labeled nucleotides, and no one really likes to handle radioactivity. So the development of that first generation sequencing methodology now used dye terminators. So instead, fluorescent dye terminators. So instead of radioactively labeled nucleotides, now we had nucleotides which were labeled green, red, yellow, and blue, 1 color per nucleotide. And now we only needed to do 1 run, just a single run. There's no need to do more than 1 run, because you can detect the different colors as they pass through the capillary, because now these were all done on capillary electrophoresis, as they pass in front of the camera. So we've avoided or removed some of those limitations. However, we still need to know the sequence. It's still important that the sequence is known, so primers can be designed for the fragment of interest. And you can still sequence a single short fragment, this time of, if you're lucky, up to 850 base pairs in a single fragment. Now, when you think of the human genome, which is 3 billion nucleotides, if you're sequencing that 850 base pairs at a time, that is going to take an appreciable amount of time. In fact, the Human Genome Project, which started back in 1990, took 13 years to complete, with the contribution of a number of big sequencing centers around the world, and it cost approximately $2.7 billion. So that was a huge cost for the Human Genome Project. A few years after the completion of the Human Genome Project, which wasn't really complete, by the way, we still missed data of telomeres, centromeres, and highly repetitive regions. But around 3 years after the completion of the Human Genome Project, second generation sequencing, what at the time was called next generation sequencing, the ideas were conceived and launched. And this opened up the genomics era, an era where now we sequence genomes on a regular basis. There are databases with hundreds of thousands of exomes or genomes which are freely available, and the price per genome has now decreased to around $1,000 per genome, sometimes even cheaper. So what's different between the first generation and the second generation sequencing technologies that made this shift possible? So there are a number of different technologies that are commonly used for second generation sequencing. There were others which are no longer used, they fell by the wayside, but what is currently used is the ion torrent technology, the Illumina technology, and the NB-seq technology. The biggest shift or the biggest change in technology was that now, instead of sequencing a single fragment in a tube in solution, we are sequencing millions of DNA molecules in parallel, and those molecules are attached to a solid surface. And this is what makes it possible to sequence them in parallel. Because instead of having sequences in a solution floating around, you have them now bound to a solid surface. You can have many sequences, many fragments bound to that solid surface, and you can track those fragments as they are elongated, as they are sequenced in real time. A second big advantage is that no prior knowledge of the DNA sequence is required, because we use adapters so that each fragment starts and ends with the same sequence. This, of course, generates huge amounts of data, and that huge amount of data needs bioinformatics to be able to put it all together again. The first step of high throughput sequencing involves fragmenting the DNA molecule into many, many, many small fragments. So think of it as a big jigsaw puzzle, which has now been broken down into pieces. Those pieces are sequenced, and then you have to put it together again. And this is why we need bioinformatics to be able to put the fragments together again, to recreate the entire genome and to identify any variation between the genome that you're sequencing and the reference genome. And just to give you an idea, the genomes of any two individuals which you care to compare, unrelated individuals that you care to compare, there are between 4 and 6 million differences between those two genomes. The technology itself has shown itself to be highly robust, to be very scalable, whereas before we could sequence a fraction of a genome at a time. Now we can sequence multiple genomes at once on the same chip at the same time, and also very accurate. We have accuracies of 99.9% with second generation sequencing. It has also been possible to apply this technology to a number of different things and research questions. We can sequence whole genomes. It can be human genomes, non-human genomes, but entire genomes can be sequenced. We can select parts of genomes and target sequence only those parts of the genome. We can use the same technology to sequence messenger RNA and therefore achieve the transcriptome. We can selectively sequence non-coding RNA. We can sequence ancient samples, and that opens up the whole field of evolutionary genomics. We can sequence organisms whose genomes are completely unknown, and I put a picture there of a local crab, which is found only here, that is being sequenced by one of the research teams at the university. And the technology can also be applied to look for methylation changes. So one technology which basically is replacing many other things which we used to do before separately. So I mentioned that there are 3 main technologies which are on the market and easily accessible. They're different in themselves, but they're also very similar. They're very similar because all technologies involve 3 main steps: a library preparation step, an amplification step, and a sequencing step. But how the libraries are prepared, how they are amplified, and how they are sequenced are different for every technology. So library preparation involves fragmentation of DNA, ligating adapters to the ends of that DNA. So every DNA molecule starts and ends with the same sequence. And if you're sequencing multiple samples at the same time, there is also the addition of barcodes, so that then the data can be attributed to the right sample. For DNB-seq, there is an additional step, which is the creation of circular DNA molecules, which are single-stranded, but I'll go into that as we go through the technologies. Clonal amplification is also different for the 3 technologies. Ion Torrent uses emulsion PCR, Illumina uses bridge PCR, and DNB-seq uses rolling circle replication, and I'll go through those as we go through the technologies. And sequencing is also different for every technology. We have ion conductor sequencing, which is pH-based for Ion Torrent, sequencing by synthesis, which uses reversible dideoxynucleotides for Illumina, and combinatorial probe anchor synthesis, which also uses reversible dideoxynucleotides for DNB-seq. That will generate the vast amount of data that a whole human genome encodes. That data then needs to be analyzed, and there are a number of different steps, which I will not go into in much detail because 1 hour is only 1 hour. However, when one is planning a sequencing experiment, one needs to ask, what is it that you want to sequence? Are you sequencing an entire genome? Is it a genome of a known organism? Is it a de novo genome? Are you only sequencing parts of that genome, maybe a selection of genes that you're interested in, or maybe only the exons of the genes? What is it that you need to sequence? Is it going to be RNA? Are you looking for epigenetic changes? Because of course, every method has its own differences. In the interest of time, and because what I do mostly is genetics, I'm going to sequence on genome sequencing mostly. So the first step, as we said, get the DNA, fragment the DNA, add adapters to the end. It's slightly more complicated than that, because when you fragment DNA, you never get perfectly blunt-ended DNA fragments. Usually you get fragments with overhangs and staggered ends. So first we carry out a process which is end repair, so fill in all the gaps. and A tailing. We add an A molecule, an A nucleotide to the end of the nucleotide, of the fragment. That A nucleotide is important because our adapters and barcodes are T-tailed. They all end with a single unpaired T. And this ensures that adapters and barcodes and whatever it is that you're adding to the end of your fragment will attach in the right direction, because the T will pair with the A, and you're sure that the orange bit fits onto the green bit rather than the purple and blue bits. So you're sure of your orientation. That gives us now our fragments with the tails we added to them, the adapters, and then we need amplification. So there is an amplification step within library preparation. At the step, one needs to decide, is it going to be sequencing the entire genome, or are we going to sequence only fragments of that genome or selected areas of that genome? For whole genome sequencing, you use the entire library. We sequence all the fragments. It's amplification free, so there are no further PCR steps, or there's no need for PCR steps except the clonal amplification, which is part of the method, of course, and therefore this reduces bias. It also means, however, that the data sets are much bigger, and that the amount of sequencing data that you can obtain per sample, per gigabase of data is less. Whereas with whole exome sequencing or panel sequencing, you need to add a target capture step, which will select the fragments of interest. Your sequencing portion, or your pool of sequencing fragments, is smaller, and so you can sequence them much more for the same amount of sequencing information. And this gives much higher depth of coverage. So depending on what the question is, one may opt for whole genome sequencing or for selected panel sequencing, depending on how much depth of data one requires for a sample. For targeted resequencing, you need to select, you need to add a selection step. So if we take exome, for example, as an example, in exome sequencing, we've got here a representation of two genes, one shown in blue, one shown in red. The exons are in color, the introns and intergenic regions are in black. And when we break our DNA, when we fragment our DNA, which we do by sonication, you get a lot of random fragments, some of them coming from exons, some of them coming from introns. And if you only want to capture the exon bits, you need to go through a step of enrichment. And in that step, the first thing to do is to denature your DNA. You get it into single strands, and then you use synthesized probes, which are complementary to your regions of interest. These probes, the hybridization probes, are all labeled with biotin. So if you mix your probes complementary to the regions of interest to your DNA fragments, you will get hybridization of probes to regions of interest, making those regions now biotinylated. Biotin has a very high affinity for streptavidin, so then the next thing we add are streptavidin-coated magnetic beads. The streptavidin and the biotin will interact together, and now the fragments you're interested in are magnetically labeled, whereas everything else is not. So then you pass everything through a magnet, and what is retained are the fragments you're interested in, and everything else is basically thrown away. And in this way, you can select which portions of the genome you're interested in. This is only possible, of course, for genomes that we know the sequence of. So for human genomes, this is possible. For genomes of other organisms, this may not be possible. But at the end of this, you have a selected capture of fragments, which you can take forward in the process. And the next step of the process is the amplification step. And there are different amplification methodologies, as I mentioned before. I, in Torrent, use emulsion PCR. And in emulsion PCR, what we get is tiny beads, which you can see over here, and micelles. And each micelle can contain within it a single bead and a single DNA fragment. That's all. It can't contain more than that. They simply don't fit. And during PCR, which happens within those micelles, the bead will now become coated by what is essentially a forest of copies of the DNA fragment that was trapped within the micelle with it. So if we have a micelle with a bead and the purple fragment, the end result is now a bead covered in multiple copies, exact copies of that purple fragment. If we have a micelle trapped, in which there are trapped a bead and the blue fragment, we will end up with a bead coated in essentially a forest of that blue fragment. And we need this step to increase signal strength. when we sequence, if you have a single DNA molecule that you're sequencing, the fluorescence it generates or the pH change it generates is too small to detect. But if you have multiple copies which are all exactly the same or generating the same fluorescent signal or the same H+ signal at the same time, then the signal is strong enough to be detected. So this is the reason why we need an amplification step. And IonTorrent solved this by carrying out emulsion PCRs within micelles. Illumina's way of tackling this amplification issue is by what is called bridge PCR. All DNA fragments, which now have adapters added to their tips, are bound to a solid slide, to a solid surface, which has a lawn of primers. Because DNA does not like being single-stranded, DNA will bridge over so that at least the adapter primer part can be double-stranded. And then the polymerase will synthesize the second chain. So through this process, each fragment of DNA will develop around it multiple copies of the same fragment. And we call these a clone of DNA fragments. The NB-seq use rolling cycle replication. In the NB-seq, library preparation has an additional step, which is circularization of the fragments. The fragments are made into a circular DNA molecule, and that circular DNA molecule is amplified many times. The polymerase will attach to one end using the adapter as a primer and will simply roll around, walk around the circle, go around the circle, giving us a very long DNA strand, a very long fragment, which contains multiple copies of the same DNA molecule. That DNA fragment, as it forms, will fold onto itself, making what is called a nanoball. So that nanoball is essentially a very long string of DNA, which has multiple copies of the same fragment, all folded up on each other. And each one of those nanoballs will be sequenced independently, and each one of those nanoballs has enough copies of the DNA to generate enough signal so that it can be detected. So at the end of clonal amplification, for IonTorrent, we have micelles with beads with this forest of fragments attached to them. For Illumina, we have clusters of DNA fragments, all exactly the same. And for the NB-seq and the rolling circle replication, we have nanoballs, and each nanoball contains multiple copies of the same DNA fragment. The next step then is sequencing. And here we have two distinct sequencing methodologies. One is a sequencing by synthesis method using blocked fluorescent nucleotides, and that's what's used by Illumina and the NB-seq. And IonTorrent use the release of an H+ molecule when the sugar phosphate bond is formed, and they detect actually that change in pH, the change in pH brought about by the release of the H+ molecule. So for IonTorrent, the method is a little bit laborious because after PCR, so after the emulsion PCR is finished, all those beads are then plated out onto a very small microchip, which has many tiny nanowells. Each one of those wells can contain a single bead with its forest of fragments, and at the base of each well, there is an ion-sensitive layer. That layer can detect the change in pH that is generated when an H+ molecule is released as the sugar phosphate bond forms. Now, since H+ released by an A, a T, a G, or a C nucleotide is still an H+, what is done with IonTorrent technology is that a single nucleotide is added at a time. So A is added to all the wells first. And wherever there is a free T on the template DNA, the A will be incorporated, an H+ is released, and a signal detected. Other fragments where the first free nucleotide is not a T, but a G, a C, or an A will not incorporate a nucleotide, and there will be no signal. After that, everything is washed away, and the next nucleotide is added. And let's say the next nucleotide is T. So any fragment with a free T as the next possible position, with a free A at the next possible position will incorporate a T and release an H+. If there are a string of A's, then a number of T's will be incorporated one after the other. So in this example, there are 2 A's coming up in the sequence, and therefore 2 T's will be incorporated within the growing chain. Since 2 T's are incorporated, there are 2 sugar phosphate bonds formed, and therefore 2 molecules of H plus are released. And this gives us a higher signal, which the computer interprets as the incorporation of 2 T's. And this is done for C and G again. And then you start over again. Add A, detect, wash. Add T, detect, wash. So it is laborious, but it's all robotized, so there isn't a person adding each nucleotide at a time, but it takes much longer because you're going through one nucleotide at a time. For Illumina, because we use fluorescently labeled blocked DNTPs, all 4 nucleotides can be added at the same time. So after clusters are formed, after clonal amplification, fluorescent nucleotides are added to the sequence. They are special nucleotides because their 3' OH is capped, it's covered. So only a single nucleotide will be added at a time. But a single nucleotide will be added to every fragment in every cluster. And once the nucleotide is added, a signal is collected. Everything is washed away, the nucleotides are deblocked, and the next round of nucleotides are added. So every cycle, every fragment grows by 1 nucleotide. And at the end of every cycle, a signal is detected, and based on the fluorescent signal, this can be interpreted into nucleotide sequence. With the NB-seq, the detection of the sequencing part is very similar. There is an anchor, which is the primer that matches the adapter, and fluorescent nucleotides are incorporated one at a time. And as the fluorescent nucleotide is incorporated, the signal is generated. The difference here is that because that nanoball contains many, many copies of the same sequence, we have this thing happening within the nanoball at hundreds, if not thousands, of different places, giving us a strong enough signal to be detected. So a nucleotide is added, the sequence, the nanoball slide is imaged, the nucleotide is unblocked, everything is washed, and the next nucleotide is added. And in this way, we generate sequencing data. Now, sequencing data can be generated for up to 150 cycles. So every one of these fragments are sequenced up to 150 base pairs. We usually have 150 cycle step. And that's pretty much as you can get out of the sequence. So if you think about it, you've got 3 billion nucleotides and you're sequencing them 150 nucleotides at a time, that there's going to be a lot of data, yes, but it's going to be very hard to put it all together. So one way by which one can get more data per fragment and can make alignment and mapping easier is by using what are called paired end reads. So single end reads is when you sequence in one direction. So here we've got a yellow adapter and a blue adapter. For ease, I'm using colors. So in single end reading, you start from the yellow adapter and you read through your 150 cycles, so 150 base pairs. If you do paired end sequencing, then you sequence again with a primer which matches the blue adapter. So now you sequence the fragment from the other end and you generate another 150 base pairs for the same DNA fragment at the same position on the slide. So with paired end reads, we get double the sequencing data because instead of 150 base pairs, we get 300 base pairs of data from each fragment. And we know that because our fragmentation gave us fragments of around 500 base pairs, that's what we aim our sonication to give us, then we know that these 300 base pairs are within 500 to 600 base pairs of each other. This helps in the accuracy of the mapping. You have 2 lots of 150 base pairs, which you know will fit within 500 base pairs of each other. The NB-seq also has a slightly different technology by which they sequence the reverse strand. Because they've got a continuous strand of multiple copies, they generate extra copies of those strands, which they call multiple displacement strands, and then they reverse sequence through those multiple displacement strands rather than sequencing again the forward strand. But both methods, though they're different, will essentially give the same thing. 2 reads of around 150 base pairs, which are tethered in the sense that you know that they are within a certain distance of each other. And this makes putting it all together much easier. Detection for ion torrent and DNB-seq, which both use fluorescence, is essentially using a microarray scanner, which will identify the clusters on the array, and through repeat imaging, an image after every cycle of nucleotide addition will capture color, and through that color it will then be interpreted into a DNA sequence. The difference here is that for the nanoball technology, the slide has wells edged in a very regular pattern, and 1 nanoball fits per well. So you don't get overlapping nanoballs, but they sit next to each other, each one in their independent wells. For Illumina technology, it's a plain slide, a clean, smooth surface, and clusters will form wherever the fragments bind, and sometimes you get some overlapping clusters, which data from which cannot really be used. It's impossible to discern when two overlapping clusters are detected together. After all of this, you get data, huge amounts of data. Depending on how much you're sequencing, whether it's a genome, an exome, whether it's 1 exome or 1,000 exomes, there will definitely be a lot of data. And that data needs to somehow be put together again. And this is where bioinformatics comes in. I won't really talk much about bioinformatics, it's not my field, but in extremely simple terms, you have your jigsaw puzzle in tiny pieces, and now you have to put this jigsaw puzzle together again. As you're putting the jigsaw puzzle together, you also need to identify places where the genome you're studying is different from the reference sequence. So we have these many short reads, and these short reads need to be aligned to the reference genome sequencing. And any differences, as shown here in this little rectangle, any differences between the reads of the sample of study and the genome, the reference genome, needs to be highlighted. So here, for example, the reference genome has an A nucleotide, whereas every one of our reads which overlap this position have a G nucleotide. So this would be called as a homozygous G variant. Now you'll notice that we don't only have a single read which overlaps this A nucleotide. Here in this example, there are 8 independent reads which overlap this nucleotide. These 8 independent reads will come from 8 different emulsion PCRs, or from 8 different clusters on an Illumina slide, or from 8 different nanoballs of the DNB-seq methodology. Keep in mind that when you do DNA extraction, you're extracting DNA from millions of cells. All that DNA goes into the fragmentation process, so every region of the genome will be in many different fragments because you started off with many copies of the genome. And because there is an error rate inherent in sequencing, the polymerase itself has an error rate. Because, of course, we have 2 alleles, 2 copies of every chromosome, then you need to have a number of independent reads which span every nucleotide of the genome to be able to call without doubt any variants. And typically, we try to aim for a coverage of 20 times minimum for whole genome sequencing. 30 times is better, but it starts getting expensive. When you're doing exome sequencing, because you're sequencing less material, we usually get coverage which is in the region of 100, 150 coverage per location. And what I mean by 100, 150 is there would be 150 fragments like these 8, which you can see on this slide, which all overlap the same genomic location. And the more coverage you get, the more confident you are of your variant calling. The more you can be sure that this is a true variant, and whether it is heterozygous or homozygous. So data analysis involves a number of different steps. And the primary analysis is what happens on the machine. Analysis of the signal, converting the signal, whether it is pH release or whether it is fluorescence, into a base, into a sequence, collecting quality data, because you need to know how sure you are of the base calling, and generating a file, a sequence file, which includes the quality data, which we call a FASTQ file. That FASTQ file then goes into the secondary analysis, which is the processing of the sequencing data. It is aligned to the human reference genome. If there are duplicates, which are usually an output of PCR, PCR can generate duplicates. Those duplicates need to be removed. If there are indels, small insertions and deletions, the alignment needs to be realigned, because where there are indels, it won't line up perfectly, and therefore you need to recheck those areas. Everything is done bioinformatically. There will be calibration of quality scores, and the output of that is a variant call file, a list of variants which are detected when comparing your sample of interest with the reference sequence. And as I said before, that list of variants, if it's a whole genome, can be anything between 4 and 6 million variants, which is a lot of variants. Some of those variants could be false positive variants, variants which come from areas which are mismapped, variants which come from areas where the quality calling is not good enough, and those are removed during secondary analysis. But then what's left, which will be a number of millions of variants, we will need to go into what we call tertiary analysis. And in tertiary analysis, you need to prioritize variants. Which of those 4 million variants are variants of interest? If you're doing rare disease work, which I do, and then you're looking for 1 or 2 variants. So out of those 4 million variants, you need to prioritize your top 1 or 2 variants, the top variants which could explain the disease that you are investigating. And then you need to validate those variants. And that validation can take the various forms, depending on what the research question was. The validation could be checking in other patients, it could be checking in a different cohort, it could be doing functional work, maybe creating an animal model. So validation can vary depending on what the research question was. I did say I was not going to go into a data analysis, but I do want to explain a couple of things about the primary analysis and this thing of quality. How sure are we of the quality of our data? So the data that's collected in Illumina, for example, which is what I'm most familiar with, is fluorescent data. So this spot over here fluoresces blue, so it's a T. The same spot in the next cycle fluoresces green, so it's a G. The next spot in the same cycle fluoresces orange, so it's a C, and so on and so forth. And based on that, a sequence is built. But besides collecting fluorescent data, we also collect what is called Phred score data, quality data. How sure is the scanner that what it saw as blue is 100% blue? Because sometimes you see you get blue and red at the same spot. So how sure is the caller that it is blue and not a combination of blue and red? So how sure is the call? And for Phred scores, we generally take a Q cutoff of Q30. So the Phred score cutoff that we typically take is an accuracy of 99.9%. We want to be 99.9% that if it's being called a T, it is really a T. And this quality data is generated by the scanner as it collects the fluorescent signal. And the computer basically expresses this as an ASCII code. So Phred scores are logarithmically related to probability of a core error. So where P is the probability that there is an error, Q, which is the Phred score, is minus 10 log base 10 of the probability. And if you take a Phred score cutoff of 10, then your accuracy is only 90%, which in a genome of 3 billion bases means you have many errors. If your Phred score cutoff is 20, then you've got an accuracy of 99%. And if you take a Phred score cutoff of 30, then your accuracy is 99.9%. And usually we aim for 99.9%. So this is what the data looks like when it comes off the machine. Each block is the data that comes out from one dot, from one spot, from one cluster. You will realize that at the top, there is a sequence of C's, A's, G's, and T's. That is the sequence. And then that is followed by a sequence of other letters and symbols and numbers, and that's the quality data. And ASCII, which I never remember, but basically this is the ASCII code. Anything which is all the way up to the greater than sign is a quality data less than 30. Anything which is question mark at or the alphabet and going upwards has a quality score which is better than 30. So what we do computationally, bioinformatically, is look at your 150 base per read and see how many of the symbols are paired with a symbol that, or how many of your bases are paired with a symbol that is question mark or better. If most of your sequencing read is question mark or better, then it's a good quality read. If most of your reads are less than question mark, so all the symbols which come before, then it is not a good quality read. And if it is not a good quality read, then that read is not taken into consideration. There are a number of things which can give rise to bad quality data, and in the interest of time, I'll just tell you what they are rather than explain what they are. But one of them is incorrect cluster boundaries. If you've got clusters which are very close together, the computer will think they're one cluster, but when in reality they're not. So as they start growing, as the chain grows, and the signal coming off the different subclusters is different, there you get two colors at the same time, and that pushes down your quality data. And another problem is a problem of phasing. As the clusters are growing, as each fragment is being elongated together, maybe a nucleotide is skipped, or a nucleotide is not blocked, or a nucleotide is unblocked and two nucleotides are added at the same time. So the fragments within the cluster will no longer start increasing at the same rate. Some are slower, some are faster. And this again gives you different fluorescent signals, which result in low quality. And this it's important to be aware of this data, because sometimes you look at reads and you go like, oh, why do I have 10% bad reads which I lost? Can't I recover anything from them? And the reality is that you could try, but what you're getting is garbage, and what you're getting cannot really be used. So QC is extremely important, and this is why I mentioned these things. And there are different types of QC. There's the Phred scores, looking for adapters in sequencing, reducing for excessive duplicates. So this is QC of the data. How good is my data? And typically we use fast QC to check how good is our data. But with high throughput sequencing, it's also important to carry out sample QC. So to check for sample mix-ups, to check whether if these people should be part of the family, are they really some members of the same family? If you know that the sample should be from a male and the sample should be from a female, are they really like that? Just to make sure that there are no sample mix-ups, especially when you're dealing with large data sets. If you've got thousands of genomes you're analyzing, then QC, sample QC is extremely important. And typically we use Plink for sample QC. The data, when it's been analyzed and it's available and you can look at it, can be viewed. So the only thing I'm going to show you is a screenshot of a viewer. And basically what you have is every pink block is a forward read sequence, and every blue block is a reverse read sequence. And when they're connected with a line, it means they came from the same cluster. So those two should be within 500 base pairs of each other. And you should have multiple coverage, many reads which fall on, which cover the same nucleotide. And if there's anything that's different, like for example here at this position where there is a red and blue line over the gray, that's because there are nucleotide differences within the reads. And you do need those multiple reads, that coverage, to be sure of what you're calling as a variant. So in summary, second generation sequencing is massively parallel high throughput sequencing, which has a number of advantages, which overcomes a number of problems we had with first generation sequencing. Mostly, we do not need to know anything about the sequence to carry out second generation sequencing, because we use those adapters to replace primers. We can pretty much sequence anything. The technology can be used for DNA, RNA, identification of methylation patterns, and everything else I mentioned at the beginning. So it is a very flexible technology. And second generation sequencing has high accuracy. There are very low error rates. The one main limitation of second generation sequencing is that the fragments it generates are extremely short. You get 150 base pairs per sequence. If you're doing paired end, you get 300 base pairs per fragment. This means that the difficult regions of the genome, repeat regions, duplications, inversions, pseudogenes, are extremely difficult to detect accurately. And it is very difficult also to carry out long-range haplotyping. It's possible, but difficult. And by long-range haplotyping, I mean if we find 3 or 4 variants within a gene in an individual, how can we figure out if they're on the same allele or on a different allele? Because that, of course, determines whether a person is, in essence, heterozygote or homozygote or compound het for variants within a gene. So the short fragments are the main limitation of second generation sequencing. And this is mostly what is driving the development of third generation sequencing. And there are two main technologies which are now commonly used. I'm pretty sure that more things will be developed in the near future. But the two technologies which are commonly used nowadays are Oxford Nanopore technology and the SMART technology of Pacific Biosciences. And whilst they're very different, and they are very different, both of them have the same key aim: to sequence single molecules at very long range. So to generate very long sequences, thousands kilobases in length, thousands of base pairs in length from a single molecule. So Oxford Nanopore technology, as the name says, uses a nanopore-based technology. Both third generation sequencing methods do not have an amplification step. They actually sequence the DNA molecule as it is. In Oxford Nanopore technology, we have pores, which you can see here in this figure. They are actually pores of alpha hemolysin, and those pores are embedded within a membrane, a synthetic proprietary membrane which ONT developed for themselves. The top part of the pore unzips the DNA molecule. So the DNA double helix is fed through the molecule, and it opens it up essentially to give you two single strands. And the second pore is where the DNA molecule passes through and the sequence is detected. Now, the thing is that within the pore, there is a continuous flow of ions. But as the DNA molecule is fed through the pore, it blocks the flow of ions. And because each nucleotide base has a different size and a different shape, the flow of ions is blocked to a different extent. So as the flow of ions is blocked, depending on how much or how little it's blocked, that can be interpreted as a base. So a T will block ion flow differently to a G, which blocks ion flow differently to Na, which blocks ion flow differently to a C. So as the molecule is fed through the pore, based on how the current through the pore changes, that is interpreted as a DNA sequence. So the unique electrical profiles which are associated with each base are used to convert that electric signal or that change in signal into a DNA sequence. The good thing about such a technology is that modified bases, so bases which are methylated or acetylated or have any other modification bound to them, will block the current differently. So in this example, we have G nucleotides, for example, here in red, and the modified G nucleotide, a G with a methyl group, also here in red. And the profiles, which are superimposed at the end of the slide, are the profile of the non-modified sequence, which is in red, overlaid by the profile of the modified sequence, the sequence where the G nucleotides have got methyl groups, in blue. And you'll notice that the profiles are different. And the profiles are different because a methylated G, the structure of a methylated G, is different to the structure of an unmodified G, for example. And therefore, the way it blocks the current passing through the pore will be different. Therefore, one other added advantage of the third generation sequencing technologies is that in the same experiment, besides detecting DNA variants, you can also detect modification of the nucleotides. So through the same experiment, you will get your genome sequence, you will get your DNA variants, and you will also get your epigenetic changes. This is possible because there is no PCR amplification step at all. Whenever we use PCR to amplify DNA molecules, then we lose all the epigenetic marks because what is synthesized by the polymerase will be simple A's, C's, G's, and T's without any modifications. Because there is no amplification step in third generation sequencing, all base modifications are retained and can be detected within the same experiment. PacBio Smart sequencing gives the same type of data, but uses a very different technology to do it. So DNA is fragmented again, even for ONT sequencing, we need to fragment DNA, but the fragments now are much larger. We get kilobases, if not megabases of DNA fragment. There is again no clonal amplification, so any base modifications are retained. And there are adapters which are ligated to the molecules the fragments that we generate. These adapters, however, are slightly different because they are hairpin adapters. So the hairpin adapters, one binds to the forward strand, one to the reverse strand, makes our now strand circular. And this is called the smart bell template. So in PAG biotechnology, we create what is called a smart bell. That smart bell are the forward strands shown in yellow here and the reverse strands shown in purple here, which are linked together by these hairpin adapters. The primers for sequencing are complementary to the adapters, and the sequencing happens because it's circular. It's very much like the DNB sequencing. We generate many copies of the same circular DNA molecule. Sequencing happens within tiny nanowells. So the sequencing chips that PacBio use are called smart cells, and each smart cell has millions of these very tiny nanowells, which they call zero-mode waveguides. Essentially, they're very small wells. Each well can hold only one smart bell, and each well is completely opaque to the well next to it. So there is no contamination or cross-contamination of signal from one well to the other. Another key difference in the PAG biotechnology is that the fluorescent nucleotides which they use have the fluorescence linked to the third phosphate group. So they're called phospholinked nucleotides. The third phosphate group is the phosphate group that's cleaved off when the sugar phosphate bond is formed. So the fluorescent dye is cleaved off as the sugar phosphate bond forms, which means that as the sequencing happens, detection can take place in real time without the need of stopping to wash away and unblock and add the new nucleotides. These zero-mode waveguides, so the tiny nanowells, have a very small volume, and nucleotides, fluorescent nucleotides, flow in and out of those wells randomly and very rapidly. So rapidly that their fluorescence cannot be detected by the system. The only way to detect fluorescence is if a nucleotide is captured by the polymerase and held in place long enough for the sugar phosphate bond to be formed. So the only signal that's detected is a nucleotide captured and held in place until it's attached to the growing strand. As it's attached, the fluorescence is cleaved off because the final phosphate group is cleaved off and the molecule is not fluorescent again. And then the next nucleotide comes in. If it's held in place by the polymerase until the sugar phosphate bond forms, the fluorescence can also be detected. So as the chain grows, fluorescence is detected only of those nucleotides which are captured by the polymerase and used for chain elongation. Now, third generation reads have a high error rate. The pair-base error rate, or rather the pair-base accuracy, is around 95%. So we're talking of really high error rate here. But with, for example, the SMART technology, because you sequence the forward strand and the reverse strand independently, and because you have what they call subreads, so the polymerase makes many copies, so it sequences each SMART bell multiple times, then you can generate a consensus read. And if a nucleotide is showing a variant in one of the subreads but in none of the others, you can say this is an error of the polymerase. But if there is a variant which appears again and again in every subread, including the forward and the reverse strand, then you are sure that that is a single nucleotide variant. So these subread passes and the consensus sequence helps to improve the accuracy. It is nowhere near as high as accuracy of second generation sequencing yet, but that's where we're going. That's where the third generation sequencing technology is going. Like Oxford nanopore, the difference or the length of time a nucleotide is held until the sugar phosphate bond is formed varies if that nucleotide is modified. So there are different kinetics for normal and unmodified nucleotides and for modified nucleotides. So like Oxford nanopore, PAG biosequencing gives you long reads and also gives you epigenetic information within the same experiment. So you can basically get two data sets in one run. You can get the whole genome sequence and all the epigenetic marks which are present on that genome. So third generation sequencing are long read, high throughput sequencing technologies. The two main ones are Oxford Nanopore and PacBio Smart Bell technology. Like short read sequences, we have the advantages where we don't need prior knowledge of the sequence. We can get the DNA sequence, RNA sequence, and methylation patterns, and we can also get DNA and methylation patterns in the same experiment. The reads now are very long. You can get kilobases of reads all in one sequence. This makes it very good for structural variant analysis. You can actually sequence across a duplication, or sequence across a deletion, or sequence enough of a pseudogene to know whether it is the gene or the pseudogene. The problems, which there still are and which are slowly being solved, are that read accuracy are still nowhere near as high as second generation read accuracy. It is still expensive. It can cost various thousands to sequence a single genome, and it requires more DNA input because there is no amplification step. So you need more DNA to actually get the data. So I will finish off with this. So if you were to visualize third generation reads, second generation reads for the same region, you will immediately appreciate the differences. For second generation reads, we have many tiny fragments, and we can pick single nucleotide variants. Every vertical line, which is a different color, a blue, a yellow, a green, a purple, shows a single nucleotide variant, but we cannot detect deletions, for example, or duplications or translocations or any rearrangements. With third generation reads, our read blocks are much bigger. You'll notice that the pink and blue blocks are much larger than the pink and blue blocks of the second generation reads. You can still pick single nucleotide variants, though the accuracy is not as good as one would hope. And if there are deletions, insertions, inversions, duplications, those can also be picked out. What you can see in this image here, over here at the end, all these reads are broken by a little narrow line, and that is a deletion. That is an area where the read starts at the start of the pink and continues all the way through to the end of the pink. But this little area, when compared to the genome sequence, which you can see here down here where it says bed file or genome annotation, that little area is missing. There is a little deletion there, which we can pick immediately with third generation reads. With second generation reads, it takes a much more trained eye to see them. You'll notice that in the second generation reads, the forward and reverse pairs are further apart across this area than they are in other places. So you could potentially pick it out, but it's much harder to pick out than third generation reads. So all the different newer high throughput sequencing technologies have their advantages and their disadvantages. What you choose, what you opt to go for, will in most cases depend on what your research question is. What is it that you are after? Are you after single nucleotide variants within a small panel of genes? Are you after single nucleotide variants genome-wide? Do you want epigenetic changes? Are you after structural variants? Depending on what data you want and what data you want to analyze, you might go for second generation reads, third generation reads. The best situation is you can afford both second and third generation reads, because they can confirm the findings of each other, and you will detect things with one technology which you cannot detect with the second technology. And with that, I am happy to discuss or answer any questions that you might have.
Wow, thank you so much for this very in-depth elaboration on different technologies. I really enjoyed to see how much we progressed in, I would say, last 2 decades. And thank you so much. So we are now opening discussion. I have many questions to ask, but I will offer others to start and then fill in. So please, are there any questions for other panelists? Maybe. We have a question for Fabio. I'm just okay. Fabio, please.
Can you hear me?
Yes.
Yes.
Okay. So thank you so much. It was a really great presentation. So I have a couple of questions that are very retain, no, they have to do with the things I'm trying to do. So it's a conflict of interest here. So first question is, what is your experience about Oxford nanopore coverage that you reached to achieve? Because we have only one try that we did, just trying to characterize in a genome editing project the site of genome linkage, and our experience was not that good, meaning that lots of money to get very few reads in return.
Okay, so disclaimer here, I have done a fair amount, or I have handled a fair amount of second generation read data. We have done far less third generation read data. Actually, I've been involved in one project, and now we're working on getting a second project going. And we had your question was like, which one do we go for? ONT, PacBio, what will give us the most data? And we've talked to a variety of people, some of them offering the service and others who have experience. And we got pretty much the same answer from everyone, that you would get less data with ONT, more data with PacBio. ONT is, if I recall well, slightly better in terms of accuracy, but you get less data. Or at least this is what I have been told, because I haven't done any ONT myself. In fact, we opted to do PacBio sequencing.
Okay, yeah, so now I know I picked the wrong technology. Okay.
Not necessarily, but yeah, coverage is an issue sometimes.
And another question is, so how good we are at determining DNA sequencing from RNA-seq data? And I'm considering this of a large amount of data sets that are there. And we know that there are somatic mutations in tissues. And so how feasible it is to use these data sets, of RNA-seq data sets, to try to pick somatic mutations and discern them from what is error of the system?
It will depend first and foremost on how much data you collected. If you want to use your RNA-seq data to call sequence, not just to look at abundance, you need a lot of data. I think we did, I don't know, Stephanie's probably here and she'll remember far more than I do. But for our RNA-seq, if I recall well, we did 100 million reads. So if you want to get sequencing data, you need a lot of data rather than the lower amounts of data which one would get if all you were interested in were quantification. And then you need, and here I don't know a thing about it, but then you need different bioinformatics packages, because the bioinformatics processing to get the sequencing data is different. So you would need to use different bioinformatics to get data, sequencing data out of your RNA molecules.
Okay, so, but you have never tried, right?
We are in the process of trying. So we've done RNA-seq, we've done we collected a lot of data, so 100 million, if I'm not mistaken, reads. So far, we've just looked at quantities, but the next thing coming up is looking at the sequence itself to identify specifically things like fusion genes. Yes, I'll tell Stephanie, if it works, we'll give you a call.
Thank you.
Thank you so much, Fabio, for these questions. Anybody else has a question from the audience? Okay, maybe a little bit later. I have a few more. Oh, okay, Yael, please go.
It's just a very short question regarding the third generation sequencing, because you mentioned the detection of modifications of nucleotides, but then I guess in the second method, so that's the PacBio, you still have base pair binding, right? So if it doesn't change the actual base sequence, you wouldn't pick up a modification, or do you?
You can if you collect kinetic data.
Okay.
If you collect but collecting kinetic data from what I can understand from our project that's going on, you need to actually tick some extra box as you're collecting data on the machine.
Yeah.
So that it doesn't just collect sequencing data, but it also collects kinetic data.
Right.
And then the kinetic data can be interpreted as modifications.
And then your bioinformatics become even more complex, for sure.
I am pretty sure it gets more complicated after that.
Okay, thank you, but that's really cool. Thanks.
So I was wondering if we can just clarify a few basic things that might be good for students to understand. These were the things that kind of confused me when I started learning about sequencing. So first, I would like to kind of elaborate a little bit more about the sequencing depth. So if you can just clarify what this term means, and just to put in the perspective, when we say 1 million reads, 50 million reads, 100 million of reads, which is considered as deep sequencing, which is considered as not deep sequencing, and if we just can like basically clarify for which purposes we can go deep, like 100 millions, and for which purposes we go for one, like if I'm a customer, just to know where to target my desires in that.
Okay, so if you're doing genome work, so if you're sequencing DNA or an exome or a selected panel, what you need to discuss with your service provider is what coverage they offer per base. So if I take this nucleotide over here, you know, at this position of the genome, whatever it is, how many reads in terms of million reads of data do I need so that my coverage at that position and any other position in the genome is minimum 30 or minimum 50? Now, this coverage that we're talking about here is how many times is that region of the genome sequenced independently. So if you're doing ion torrent, how many of those micelles contain the fragment which comes from that region? If you're doing Illumina, how many of your clusters contain that region in a fragment? If you're doing the NBSeq, how many nanoballs contain a copy of that region in the fragment? So how many independent reads? And this is one of the beauties of high throughput sequencing. With Sanger sequencing, you just get everything in one solution and you get one signal. Here, every single read, every one of those clusters, nanoballs, micelles, everyone is sequenced independently. And if you look at this IGV snapshot, every one of these lines, this one is data from one cluster, and this is the data from another cluster, this as well. So coverage here is something like 30. We have around 30 different fragments from different places on the chip, which include a DNA fragment that spans that region. And the more you have of these 30, 50, 100, 150 reads which span the same region, the more confident you are of your variant calling. So it's a simple question of numbers, right? If you have coverage of two reads which span a region, and both reads are A, what will be your conclusion? Your conclusion is that this is homozygous A. But if you then get a third read, and the third read is a C, suddenly you've got a little bit more data. So what is the genotype at that position? Right. And you might say heterozygous C. Okay, AC. And then you get another 5 reads and they're all A. So now you start thinking, oh, is that C a mistake, a polymerase mistake, a chemistry mistake? And through experience, whenever we tried to handle data where depth of coverage was less than 20, and then we tried to confirm using other methods, essentially Sanger sequencing, it didn't always confirm. But when we took a cutoff of a minimum 30, and preferably more, unless we're dealing with pseudogene regions, which are their own nightmare, which I won't go into, then it confirmed every time. So depending on whether it's a whole genome or part of a genome, and depending on what depth of coverage you want, that is how you calculate how many million reads you want. And it will vary from experiment to experiment because it depends on how much your sequence is in terms of millions of bases. Is it whole genome, part of a genome? Is it human? Is it mouse? Is it fish? It's going to vary like that. And how much depth you want. Typically, what's offered for a whole genome is a sort of minimum coverage of 30-fold throughout, which will mean that you will have more than 30 in many places, but there will be places where you're at 30, 29, thereabouts. But it will depend on exactly what you're sequencing.
Thank you so much. So now I will transform this into more concrete situations. So, for example, I'm interested in discovering cheap clonal hematopoiesis of intermittent potential within certain samples. And we know that these, let's say, mutations are relatively rare. So how deep we need to go in order to find them on certain genes, because we are only looking at certain genes like TET or DNT, DMMT or so on. So
this is clonal, right?
Yes.
I don't know. It is something I would need to, I mean, I probably, if I asked someone who does oncology, they would know, because they are also looking for somatic mutations, and they're looking for driver mutations and for carrier mutations. And what I typically do is inherited diseases. So there you're looking at a 50% bias, or at a 50%, if it's heterozygous, 50-50. You would need, intuitively, I would say you would need a lot more coverage if you're looking for those, because it also depends. When something is clonal, it depends on how many of your cells actually arise from the clone you're after. So the rarer the clone, the more coverage you need. And the more common it is, then the less coverage you will need. At one point, you have to take a decision of like, okay, I'm happy to detect if I'm down to 1%, or no, listen, I want to go down to 0.1%. But the lower you want to go, the more data you will need. But the best person to give you an answer to that is someone who's used to handling oncology data, probably, because they are the ones who are used to this mix of reads because of clonality.
Okay, one more practical question when it comes to DNA sequencing. What types of extraction methods are mostly recommended, and what are the quality control recommendations for DNA sample to be used for if we are going for next generation sequencing?
So it depends whether you're going for second or third generation sequencing, and it also depends on whether you're sequencing, a human genome or a non-human genome for which there is no reference sequence. So you need high molecular weight DNA. You need it to be as intact as possible with good 260-80 ratios. And you have to make sure that it is not in a buffer which has high salt. For example, for us, many of our DNA samples were in the standard TE buffer, and we had to repurify everything to put it into a low TE buffer. You will, for second generation sequencing, you will fragment that DNA. So it is slightly more permissive because you're breaking it up anyway. If you're going for third generation sequencing, however, the more intact your DNA is, the more high molecular weight it is, then the easier it will be to get longer reads. There are other things which are up and coming, which use optical mapping and these really, really long DNA molecules, and there you will need ultra high DNA molecular weight DNA. We, for both first and second generation sequencing, we have managed, for second and third generation sequencing, we have managed to get sequencing data from high molecular weight DNA. For third generation sequencing, we were told if you could provide ultra high, it's better, but we couldn't extract ultra high molecular weight DNA. This was a project with a non-model organism, but it worked well with high molecular weight DNA. We typically use a modified version of the salting out technique, which is manual over 2 days, but gives us very good yield, even from smaller amounts of blood, and very intact DNA. However, I do have colleagues who have sequenced samples from, like QIAGEN and other similar kit extractions, and it worked well as well. The important thing is that your DNA is not fragmented, that you have good 260-80, good 260-30 ratios, so that there isn't carryover of either proteins or solvents or things like that, and that it is in a low salt buffer.
Okay, thank you. These are all practical recommendations that we really need to know. If I mean, it's bad to isolate everything, extract, and then your
and then suddenly you're like,
say, yeah, they say it's not good.
it is always good to, especially if you're, if you have the luxury that you're still starting the collection or the extractions or whatever, it's always good to have a good chat with the different providers, and everyone will have their own requirements, but you will see certain things which everyone asks for. And then you go like, okay, this is my basics. I have to make sure that I have this at least. And then you go on from there.
Okay. Okay, perfect. Thanks. Do we have any more questions? Paolo, please.
Just a comment. First of all, thank you very much, because this was a very wonderful primer for many of us to really try to see the details and the advantages and disadvantages. And of course, you say if you can get access to both second and third might be better, but of course, everything. Now, I was kind of, yeah, I have a quite kind of naive question, which is, I was impressed by the fact that with third generation, you can also have some, epigenetic information, methylation, and so on. Is this actually a strong advantage in comparison with the cost or other techniques, or is this a big plus or not, in your opinion?
The thing is this, it depends on the number, in my opinion, which might be very different to what other people think. In my opinion, it will depend on how many samples you need to process, what sort of budget you're looking at, and also what sort of epigenetic data you can get or you want to get, because there are other methods like Hi-C, for example, or other similar things that can be used to get epigenetic data, but you would have to do a second experiment. You would need more material, there's additional cost and whatnot. The third generation sequencing technology appears to give you that methylation data, but there is again then the less accuracy, because with third generation sequencing the accuracy is lower. And I am at this point in time, and as the third generation sequencing methods develop further, and they have developed a lot in the last 2 years, so I'm expecting them to develop even more in the coming few years. As the technology develops, that the data is more robust, more reliable in terms of accuracy, I would probably be tempted to go for that. It costs a bit more, but I get more data. At this point in time, because what I look at mostly is genetic variation rather than epigenetic variation, I would prefer to go for second generation sequencing for the bulk of my samples. And then the ones where we can't figure it out, we think maybe it's a structural variant and epigenetic change, though that's a smaller number of samples, then I would do third generation for those. And I have my second generation data, so I can compare, and any variants called, I can see, were they there in my second generation data? Yes, they were, then they're true. No, they weren't, then maybe I need to do Sanger sequencing to double check, because I'm not trusting them. So at this point in time, I wouldn't go for third generation sequencing, because I think the accuracy can and will be improved more. But if you talk to me in 6 months' time, that might change.
This is a very, evolving world, evolving area. So yes. Thank you very much for this in-depth suggestions and comments and experience. Very, very nice, very nice. Thank you very much.
And also remember what you need. If you need structural variants, the second generation data you will struggle to get them from there. So if what you're looking at is structural, then just go for third generation. So it will depend very much on your research question.
Thank you very much.
I can also confirm that Oxford Nanopore technology, at least from the perspective of RNA sequencing, is rapidly improving. Although there is not much people using RNA sequencing. We went in Maastricht on Oxford Nanopore Day. There was around maybe 150, 200 people in the in there, and only 3 people were using Oxford Nanopore for RNA sequencing. They are now producing the 4th generation of regions, and we will test it tomorrow, because with the 2nd generation for RNA sequencing, it was quite a mess if you want to do direct without the qPCR or without the polymerase reaction. But we will see the number of reads. It's quite lower. You need a vast amount of RNA. For example, we run 3 micrograms of total RNA, and we only got in 17 hours of run, we got around 1 million, slightly less than 1 million reads. Of course, these numbers of reads are not really comparable to the numbers of reads if in Lumina. This is what I was told by bioinformatician, but it's quite different. We will see what will happen with this new generation of regions. But yes, it's quite a challenge, and it's quite expensive also, because basically one sample can be run on 1 flow cell, and the price of 1 flow cell is 900 euros. So without any additional chemistry, with just the flow cell. So it's quite pricey. Yeah. Of course, it allows to detect certain amount of modifications for MSCA. Particularly now they are emphasizing this part, but yeah, it's quite a challenge to and it's not easy to optimize the methodology and so on. But as you mentioned, probably in next year or so, the technology will be much more stable. I think that for DNA, at least, it is more stable than for RNA, but probably they are improving their accuracy in every 6 months. So I think they are on a good on a good trajectory too. But currently, you say Q score 30. For us, it's around 10 currently with for the reads that we run. So it's quite different. It's basically 10% difference in accuracy, which is quite surprising.
It's because the technologies are built for different things.
Yeah.
Or at least their strengths are different. If when 3rd generation technology gets anywhere close to 2nd generation technology in terms of accuracy, then none of us will be doing 2nd generation sequencing anymore, essentially.
Thank you. Okay, do we have any more questions? I don't see, I don't know if Fabio raised his hand again the 2nd time, or it was the 1st time. No, okay. So I wish to thank Rosienne for taking this time and giving us this beautiful lecture, and of course the discussion was pretty fruitful. I just want to inform everyone that our next webinar is going to be in January. Yael, you are up next. Did we say around 25th? What was the date? Something like that. Yeah, I will check and then 22nd. Okay, we will send the invitation, of course. I wish to thank everyone all the best in New Year's. Give it yourself a good rest during the holidays. Don't eat too much. This is also for me. Just try to enjoy as much as you can, because we know then academia is quite overwhelming. So with all the things that are ahead of us, and I'm happy to see you in the next year, of course.
All the best, everyone.
Thank you very much, Rosienne.
Thank you, Rosienne.
Bye-bye.
Bye.
Thank you, bye.
Bye-bye.
All the best. Happy New Year.
Bye-bye.